Uh, okay, so good afternoon or good morning, depending on where you are. Thank you for joining us today. On behalf of the Columbia Global Center and the Institut Pasteur, I would like to welcome all of you in this 22nd lecture of the Logical Reasoning in Human Genomics course. So today we have a very special guest and speaker, and I will give the floor to our colleague Joe to introduce Professor Nabil Enattah. So the floor is yours, Joe.
Hi, yes. Good morning and welcome back to our course on logical reasoning in human genetics. Today we have a special guest. He's an old friend of mine when he was a student in Helsinki in Finland back in late 90s. And we worked together then on the project he's going to talk about today, which is about lactase non-persistence, or lactose metabolism in humans. And Nabil was the first guy that identified the variant that was responsible for all this, and one of the few actually useful genetic tests that exists in the world was developed by Nabil for lactase non-persistence. He's also been a dear friend of mine. We were my colleague Jolie and I were supposed to go to Libya in 2011 to teach a version of this very course. And unfortunately, that was delayed because of the civil war that erupted in Libya and has been going on for the past decade. But we've stayed in touch, and Nabil was actually the impetus for us starting this program with the Global Center in Tunis. I initially went there in January 2020, trying to figure out a way that we could start up our project again to help Nabil prevent brain drain from Libya and keep science alive there in academia. At the time, he was president of the University of Tripoli, perhaps the most successful student of Lena Palotia, who was our colleague back in Finland, having made it to such high levels in his country. So I just want to turn this over to Nabil, who's a very good scientist, a wonderful human being, and a dear friend of mine. And we want to hear him tell the story about lactose and also something about what it's been like to work in Libya during these past 12 years. So Nabil, we turn it over to you now.
Thank you, Joe, for the introduction. As I mentioned before, I would like to talk about the lactase project in general as a story. We had some lessons to remember. Our focus of our presentation is on congenital lactase deficiency and the second disease, or the second conditions, lactase persistence. So let's begin with congenital lactase deficiency, which is autosomal recessive disease characterized by watery diarrhea during the first 10 days of life. So it is the most severe form of lactase deficiency. It's almost total lack of lactase activity on jejunal biopsy. And if the newborn is not treated, it will suffer from dehydration and can be severe, and it can be lethal also. This disease is one of the called the Finnish disease heritage. It is several diseases which are enriched in Finnish populations due to founder effect. At that time, we had collected 19 Finnish family, 38 patients, and we worked on the family. In the beginning, we made the initial candidate region approach. Lactase gene was assigned to chromosome 2, and defected enzyme is lactase, so it was the logic to make a candidate gene approach. And we make linkage study on the chromosome 2, where the lactase gene resides. So I remember it was a clear hit from the beginning. So we had some marker, which is allele, specific allele 2, present in 92 of the disease allele. This is the close to the lactase gene. And we made extended CLD haplotype around the regions. This is the markers. And we spent many months, actually, 1.5 years at that time, trying to make even physical map on the regions. During those days, it was not easy, but the lessons, the genealogical study, there is very good genealogist, Tebo Varelo. It's a very good, really Finnish scientist. He's specialized especially in the genealogical studies. In Finland, most of the populations documentations goes back to the 16th century, so it was able to trace all the patients, and the CLD mutations was enriched in sparsely populated eastern and northern Finland, and as I said before, because of founder effect. The haplotype analysis indicate to us that there is a novel genes causes CLD, because our extended haplotype analysis has been locate the critical regions little bit far away from lactase gene. And this convince us that we are dealing with a new gene. Because the lactase genes was outside the critical CLD region. And based on that, I remember it was my colleague Mikko took the CLD project, and I began at that time to work on lactase persistent conditions. This is the 2-point Lod score, and why I'm talking, because we used the formal linkage analysis, and we had the highest Lod score between the marker D2S314 and D2S2385, and we built, this is the physical map, it's BACs and YACs around the regions. So we were very convincing that the genes, it's not the lactase, because of one mistake we have done. We misunderstand the extended haplotype analysis, and at the same time, there is a report in American Journal for Human Genetic Supplement. It was abstract published by Italian group, and they analyzed one of our CLD patients. It's a Finnish CLD patient. And they report they have sequenced the lactase gene, and they find no sequence variant within the genes, which make us to convince that there is a new gene underlying congenital lactase deficiency. Mikko has worked about 5 years trying to screen all the candidate genes in that region. And suddenly I remember one of our colleagues, he didn't find anything. He said to him, why you don't go to the lactase gene and sequence the CDN of the lactase gene and see? He got the message, he did that, and surprisingly, we find 5 distinct mutations in the coding region of the LCD gene. We spent really about 5 or 6 years working hard, and the gene was in front of our eyes. It's a very typical case for trusting in the publishing data, and it was very painful listen actually. At the end, we find it is the typical case of Finnish disease heritage. There is 27 patients out of 32. They have mutations designated Finn major, and the others for rare mutations. Most of them are really truncations, so it's stop codon. So This is the CLD haplotype after reshaping the extended haplotype analysis. And this mistake teach us that really sometimes the simple approach, it should be tried before we invoke, let's say, the difficult explanations. The lactase was in front of us, but because of some mishandling of haplotype analysis and trusting in one of our colleagues in Italy, led to this dilemma. So let's go now to the lactase persistence. Lactase persistence, it's not a disease. It's there is a lot of terms using and confusing in lactase or lactose intolerance terms. The adult type hypolactasia is a term also used for decreasing of expression of lactase enzymes after weaning in mammals. So there is physiological decline. It's autosomal recessive. The lactase persistence is the mutation or is the mutant phenotype. But during the study of these conditions during the 1950s, it was in Northern Europe. So they reverse the approach. They think adult type hypolactasia is the disease, but it turned to be it's the normal condition. That's why lactase persistence, the disease or the beneficial mutation is dominant to autosomal recessive adult type hypolactasia. The sequence analysis of the coding and the promoter region of LCT gene, lactase gene, revealed no DNA variation correlating with lactase non-persistent persistent phenotype. Here, the data was showing us to seek other approach. The linkage analysis, we made it again for adult type hypolactasia or lactase non-persistence, and the signal appear again that this is the region which is harbor the mutations for lactase persistence non-persistence, and we go through the same approach. This is 9 extended Finnish family, collected and phenotyped by lactose tolerance test. So it's verified. We use linkage disequilibrium and haplotype analysis for this 9 extended Finnish family. We are able to restrict the locus to 47 kilobase interval. This 47 kilobase, it is adjacent to the lactase gene. It is, most of them, it's located within the adjacent gene MC6. Yes. We took 4 samples from with lactase persistent and 3 samples with lactase non-persistent, and we decide at that time that we will sequence the whole region. It was visible at that time. It's not an easy, but it was very visible. We amplified overlapping PCR product, and we went through all the variants, and at that time it turns to be there is 2 variants stand out very clear to correlate with the lactase persistence phenotype. The one variant is the CT -13910. It's located roughly 14 kilobase upstream from the lactase LOX. It's completely associated with biochemically verified lactase non-persistent in Finnish family, and also in sample set, random sample, 236 individuals from different populations. And this is also the physical map we have built over the regions. And this table shows the linkage and the linkage disequilibrium in the family with the lactase non-persistence. and we can see that it is almost the same picture, very similar to what we have got with congenital lactase deficiency. There is a second variant, as I mentioned before. It's 8 kilobase telomeric to the first variant. It's also in complete linkage disequilibrium with the first variant, but there is some cases which is we found it doesn't correlate with the phenotype, so we exclude it as causing a variant, and we stress that the only variant which is correlate with the lactase persistent is the CT minus 13910. We analyzed this variant in many samples. In this study, 1,047 samples, and all the samples we analyzed, the frequency of this variant is very correlating with the frequency of lactose intolerance among those populations. Even it was most of them random samples, but it gives an indicator that this SNP, it is really a causing variant. Then we go to study this variant in 37 populations to see what is the happening, especially this condition is it contained the highest signal of selection in the human genome. So it was puzzling why this condition is so prevalent in some populations, especially with the populations which has had history of daring for a long time. We typing several SNPs around the regions in about 30 kilobase, and we try to construct the haplotype. We saw that there is one haplotype is present in all the population analyzed, which is the associated with lactase persistence, and there is other haplotypes associated with lactase persistence, but they are originating in some geographically restricted population. They are living west of Ural and north of Caucasus. That's why we make a hypothesis that the region, the lactase persistent mutation are rising several thousand years ago on that region, and from that region it's distributed west and north, and it is a very clear example of genetic adaptation to the environment. We built a median joining network using the network program, and as you saw here that there is several haplotype. The major haplotype of lactase persistence in green, and lactase non-persistent haplotype in yellow. And I have to just now concentrate on haplotype 84, we name it 84, and haplotype 98. Haplotype 84, it is a lactase non-persistent haplotype because we had verified South Korean sample, and it was in high frequency in many populations in East Asia, like South Korea, China, and the major haplotype of lactase persistence, that's the mutation, it's reside in one haplotype extending almost 2 megabase. And what is the difference between these two of them? It is almost the same size, 2 megabase. The only difference between them just the 2 mutations of lactase persistence. It can be invoked by gene inversions, but I think this is the background of haplotype, which is lactase persistence mutation arose on it. And if you see, so there is different populations having different haplotype harboring lactase persistence mutations, which is on the upper left, like Moscas, Odmor, Erzas. And these mutations convince us that lactase mutations has been arising several times during history, and there is convergent evolutions of the condition. We were tricky at that time to convince that really it's still ongoing conversions for these mutations. We make estimations for the most recent common ancestors for the mutations for these alleles, the most common alleles, and most of our estimate it's between 5,000 to 12,100 years old. The others between 1,400 to 3,000 old. So our data about the global allelic haplotype imply that the T allele independently introduced more than once, and that there is a still ongoing process of convergent evolution of lactase persistence in a human allele. And this, the tables show the 37 populations and the allelic frequencies of the lactase persistence mutation. This figure shows perfect, almost perfect correlations between the frequency of lactase persistent trait as measured by lactose tolerance test or disaccharidase activity and the frequency of lactase persistent as we predicted from the CT allele, assuming a Hardy-Weinberg equilibrium. It is the R value, it's almost 0.973, so it is really very high correlation. This is the estimation of the age of these mutations in many of the populations we have studied. And as I said before, it is almost between 4,000 to 8,000 years ago. And many studies which studied ancient DNA, they show no evidence of, so it gives indications that really the lactase persistent mutation, it's arose 3,000 to 4,000 years ago. But what is the mechanisms which make to rise in frequency in some populations, 70 or 80%, and make a sweeping? It is the selection coefficient of these conditions. It considered one of the highest coefficients for any conditions. It is still wondering. There is invoked several hypotheses, but we can't say many mutations in different maybe populations, which is arise independently, and the regions is really still in convergent evolutions, and it is a very good example. It is a very good example of gene environment interactions. And what we have done with the Saudi, because it was, I remember, it was random samples. So we took it, and we took the phenotyped Koreans, and we analyzed the same with the same haplotype. we analyzed all the regions, and we found just 2 mutations stand out, and that is the approach we made to confirm at least we are dealing with lactase persistence mutation. The high selection coefficient, almost 4% on average, but maybe in some populations, the coefficient can go even to 9 or 10%. What is beautiful here, that the European mutations and East African mutations 907, they are sharing the same ancestral background in which the mutation has been arised, and most likely they have the same history. And we think probably related to the same cattle domestication event. In contrast, the Arab alleles show different, highly divergent ancestral haplotype. It suggests that these 2 major global lactase-persistent alleles have arisen independently, and the Arab, maybe it's in response to camel milk consumptions. So there is a camel domestications happen in Arabian Peninsula about 4,000 to 5,000 years ago, and our result, it supports really the convergent evolution of lactase persistence in diverse populations. And this is the table for genotype frequency of the TC910 and the other one, which is a little bit in other exon or within in other intron, sorry, of MCMC6. It is the same like what we found in the Europeans. There is 2 mutations far away, about 8 kilobase. Even here, the region between them, it is almost 9 kilobase. But the other ones we have trying, it is almost not perfectly correlating with the lactase persistence, but maybe they are really working within the functional domains between them. Their rules need to be verified very well before to exclude it totally that it has nothing to do with the lactase persistance. And this is the regions of haplotype extended analysis. And this is the MG network, which is really very nice, shows that the haplotype 98 and haplotype now, this is the Eastern African and this is the Europeans, and most probably are sharing the same, at least European, but the Eastern African, they have both cattle and camel. And the Arabians on the left, it's most likely it's with camel domestications, and this is it is so beautiful depictions of the history of lactase persistence. And this is also for the other. So we are trying now to move for study of genetic variant of lactose among Libyans. When I came back to Libya, I have supervised almost maybe 20 master students. One of them, it's work on the lactose intolerance, and we collect in Libya 242 samples, and we found the 2 mutations, the Europeans and the Arab mutations. So the Europeans, this is the distribution. We divided on West region, South region, East regions, and it is the frequency, it is not very high, 6% in the West and 5% in the East, the least. And this is the Arabian mutations, which we call, it is between the West 20% and in the East 25%. the result, it goes very well with the history of Libyan tribe. Most of the tribe in Libya and eastern regions, they are originally came from Arabian Peninsula, especially during the 10th century and the 11th century. There is a big immigrations of two big tribes, Banu Hilal and Banu Salim. So they came to Egypt, and then they came to eastern Libya, and they passed through Libya, and they invaded actually Tunisia and controlled Tunisia at that time. So the mutations, the Arabian Peninsula mutations, it is very good indicator for the origin of those tribes. And during the sequence of Libyan samples, we found other rare mutations around the Europeans. And most likely they have, we cannot say for sure, we need a functional study to see the role in lactase persistence. But as I said before, any mutations in the physiogeny of European mutations most likely have role in lactase persistence. And this is the frequency. It is there is TA, and this is the European, this is the Arabian, and this is just six base pair, and this is almost 40 base pair, almost around. And this data, it is really, now it's a little bit old. Unfortunately, it's not published yet. And that's one of the major difficulties we face here. There is really reluctance in publishing our data in the speed we should do it. This data is very good and it should be published, but it's now keep it as a master thesis project, and I hope the student can publish it very soon, because really it is good, and it adds at least to the picture of lactase persistence in Africa or in Arabic country. During this, we found really even, we made a lot of collection of data about many symptoms and associations. We found a clear association between abdominal symptoms and lactase non-persistence with the Arabian mutations. So it goes very well with at least what we know about this condition. About selection. The selections is really intergoing for lactase persistence. There is a hypothesis, cultural hypothesis, which means that the lactase persistence arose in correlations with pastoralism, with dairying practice. And there is another hypothesis, it is a calcium hypothesis, calcium absorption. There is some benefit from lactase persistance, but as you saw here that the risk of malaria, the selection coefficient 4% to 9%, skin pigmentation European, hypoxia and response to high altitude in Tibetan population, and alcohol dehydrogenation. And also there is ectodysplasia receptor gene, which involved in hair follicle. All this high selections coefficient reported, the lactase genes is still intertwined. I'm still thinking now more than 20 years. What is the major reasons which make really this frequency of lactase persistence driven to high frequency in many populations? It's really intertwined. What the lesson had to keep short, from lactose story, you should have high suspicion index in the result of other, and you should verify it by yourself, because really it cost us several years of useless work. This is the CLD case. In the lactase persistence, when you decide to collaborate with other, please make the deal clear, written and signed. You may need a lawyer to keep you right. It is really the story is a little bit painful for me as a person, as a scientist, but that is the life. That is the life. It is okay. And I would like now to just to say thank you, and I would like to give opportunity for people if they're trying to ask about this or about the situation in Libya and the scientific research in Libya, at least during the last 12 years. Thank you.